Good evening, everyone. My apologies, my humble apologies. I'm 4 minutes late. So if you can hear me or see me, please can you give me a thumbs up just to check whether audio video is working fine or not. Can you give me a thumbs up? My apologies, my humble apologies. I'm 4 minutes late. Good evening, Shivani. Good evening, Jeet. Okay, so you can hear me, right? Because of some technical issues, actually, we are late by 5 minutes. So let's start our session, not to waste our time, right? This is regarding the FMGE session. I welcome you all and thank you very much on behalf of eGurukul powered by DBMCI. Myself, Dr. Varsha Gupta. I'm your biochemistry faculty. And in this session, I'll be going over the previous FMGE questions, right? As it is well said, if you know at least the questions which already came in the exam, then that gives you a guideline which topics you need to prepare and what you should be focusing or for the examination point of view, right? So you have a roadmap to work upon. So in this session, I'll be going over the questions and then I'll be touching all the important points related to that topic, because definitely they are not going to repeat the question as such, but relevant things they may give you, number one thing. Second thing is, since the trend is being changing, right? I have seen the MCI, they had made like lots of changes in the INI AIMS exams, even for the FMG people, right? They have made lots of changes also. So I am expecting that this time exam is going to be a little like the bar of the exam will be on the higher side, right? Thoda difficult, they are gonna do it for you, right? So be prepared, just learn little bit extra for every topic you are going ahead, right? Biochemistry is worth 16-17 points for you guys, and I'm pretty sure if you will attend these kind of a session, the biochemistry will be like an easy job for you, and that will be a like piece of a cake. If you have any questions or any doubts regarding the session, or if you want me to incorporate any particular problem of yours, you can email me, you can telegram me. My telegram is @Dr. Varsha Gupta. My email ID is up on the screen, and you can drop me an email or connect with me anytime, right? So hey Taj, hey Mulai. Good. So if you like have trouble like getting me in English, just let me know. I'll use the Hindi words in between also, right? So first question is about all the sulfur containing amino acids. All are sulfur containing amino acids. So we should know about the amino acids classification for the exam for sure, right? Amino acids, we know we can classify based upon various criteria, based upon whether nutritional, whether they are essential, non-essential amino acid, based upon their side chains, right? Based upon whether they are going to produce the ketone bodies or they are going to participate in the glucose kind of a kind of a pathway. So three major classifications we always talk about the amino acid, right? First classification is based upon the structure. We can classify aliphatic amino acid. Aliphatic amino acid, what we have, the simplest one is the glycine amino acid, is the simplest amino acid, and your branched chain amino acid. Alanine is also an aliphatic amino acid. Then I love Varsha ma'am. So I is isoleucine, leucine and valine. These are your branched chain amino acid, which I'm categorizing under aliphatic amino acid. Hydroxyl group containing amino acid. How many we have? We have 2 hydroxyl group containing amino acids, serine and the threonine, right? And the sulfur containing sulfur containing amino acids, we have the methionine and we have the cysteine, right? Acidic amino acids, we talk about the glutamic acid, aspartic acid, glutamine, asparagine. Right? Aromatic amino acid, we have 3 of them, right? Phenylalanine, right? From phenylalanine we can synthesize the tyrosine, and then another one is the tryptophan. Basic amino acid, what are basic amino acids? Very, very important for examination point of view. Basic amino acid, the biggest, most basic amino acid for us is arginine, okay? Arginine is the biggest basic amino acid for us. Then is the histidine and the lysine. And the amino amino acid, this is not the amino but the amino, this is kind of proline fold itself to make a cyclic kind of a structure, that is amino acid. Next category is based upon the nutritional requirement, whether they are essential, semi-essential or non-essential amino acid, right? Essential amino acid is, again, I love Varsha ma'am, your branched chain amino acid, right? And what? And your aromatic amino acid, phenylalanine and the tryptophan. Why we are not taking the tyrosine? Why we are not taking the tyrosine? Come on, why? Why? Because from the phenylalanine, if we do the hydroxylation of the phenylalanine, we can get the tyrosine, okay? Hydroxylation of the phenylalanine will give us the tyrosine. That is why tyrosine is not an essential amino acid. Similarly, the methionine, from the methionine we can synthesize the cysteine. That is why methionine is essential amino acid, whereas cysteine is not an essential amino acid. Threonine is also an essential amino acid, right? Semi-essential amino acids are arginine and histidine, right? They are specifically important, they become essential during the period of growth and also during the pregnancy sometimes, right? And rest all are non-essential amino acids. So we should be knowing their classification, which amino acid is what, right? Now the third classification is based upon metabolic fate also, ketogenic, glucogenic, or whether the amino acid is ketogenic, glucogenic, both, okay? Ketogenic, Liu lie, okay? Liu always lie, right? Liu always lie, just remember this way, Liu always lie, right? Leucine, lysine, right? They are the ketogenic amino acid. And for the glucogenic and the ketogenic amino acid, remember the aromatic amino acid, your phenylalanine, your tyrosine. PIT, remember this, right? Your phenylalanine, your tyrosine and your tryptophan, and is isoleucine, right? These are they are both glucogenic as well as ketogenic amino acid, and rest all are glucogenic amino acid, right? So coming back to your first question was what? If I go back, sulfur containing amino acid is what? Sulfur containing amino acid is your methionine has sulfur, cysteine has sulfur. If you join 2 molecules of cysteine together, right? Cysteine is joined together with another cysteine. They have what kind of a linkage? They have a sulfhydryl linkage, right? That makes the cysteine. So all these 3 are sulfur containing amino acid except the leucine, which is not a sulfur containing amino acid. Is there any problem in getting my voice or video? Mulay, you said buffering. So is there any issue on my part or your internet is not working well? Question number 2 is from the choice given, which pair is the source of nitrogen in the urea cycle, right? We know when we do the amino acid degradation, right? Amino acid degradation leaves us with 2 parts. One is the carbon part, right? One is the nitrogen part. We have 2 things. Nitrogen is released in the form of the ammonium ions, and the ammonium ions, or you can say ammonia, you can say it exists as an ammonia first, then it is converted in ammonia ion. It is toxic in nature, so it goes into the urea cycle, right? Where what is formed? Urea is formed. Urea is C double bond O, and on both sides you have ammonium group, right? C double bond O, left, right, NH2, NH2. That is urea, and that is transferred to the kidney, right? And it is eliminated via urine, and this is less toxic. This is less toxic. Okay, thank you Mulay, thank you Ariful. Hello Dr. Sukhvinder Singh. Okay, so this is the how we take care of the nitrogen, and the carbon part, right? It goes towards the gluconeogenesis, or it forms a part of your carb cycle. Okay, this is what is going on. Now question is about asking you which is the source of nitrogen in the urea cycle. So this is your urea. This is your structure of the urea, right? One nitrogen is coming definitely from the ammonium ions, right? And from where this second ammonia, this N is coming from, nitrogen is coming from, this is coming from aspartate. This is coming from where? This is coming from aspartate, and this carbon comes from the carbon dioxide. So these are the donors for the nitrogen and carbon in the urea cycle. First point, you should be remembering this for examination point of view, right? What else you should be remembering? The urea cycle rate limiting enzyme. Rate limiting enzyme is CPS1. Carbamoyl phosphate synthase 1 is the rate limiting enzyme. There is second enzyme OTC, ornithine transcarbamylase. Deficiency of this enzyme is important, but this is not a rate limiting enzyme. So there are few things you should always be knowing about the cycle, right? And the urea cycle is, how many steps are there in the urea cycle? There are 5 steps in the urea cycle, right? First 2 steps, they takes place in the mitochondria, and then rest 3 steps takes place in the cytosol. You should be knowing that also. Whenever you will see any cycle, any pathway, right, the starting point will always be the mitochondrial. Then it may go outside, the products that are forming in between the pathway, that may go outside and they may join back. So initially first step is usually the mitochondria, remember that. But in the urea cycle, first 2 is in the mitochondria, and the rest of the half is taking place in the cytoplasm. What else we should be knowing for the urea cycle? We should be knowing the different names of the urea cycle. Right, urea cycle, most importantly, there is this substrate that is formed in between, ornithine. That is why it is also known as ornithine cycle, right? And it is also known as Krebs-Henslate cycle. Henslate cycle. Krebs cycle is your TCA cycle, citric acid cycle. This is Krebs-Henslate cycle. These are the name of 2 scientists who discovered the urea cycle. So these are the few points we definitely should be knowing for the examination point of view, right? Now the next question, question number 3. Nitric oxide can be synthesized from which amino acid? First of all, the formula of nitric acid, it is just NO. NO, where do we see it? We see it in the form of a vasodilator, right? We see it in the form of a vasodilator. How do we synthesize it? We synthesize it. It is synthesized in the body by the arginine. Very, very important question, right? By the arginine. Very good, Ayushi Gupta. Very good. So you are well prepared, right? How do we do it? We should be knowing other points of it. Can you tell me the name of the enzyme which helps in the synthesis of the nitric oxide from the arginine? What is the name of enzyme? Can anybody tell me the name of the enzyme? Yes, Govra, you are right. Dr. Sukhvinder, yes. Which is the enzyme here. Enzyme is NOS, nitric oxide synthase, right? And there are three kinds of nitric oxide synthase: eNOS, iNOS. These are the isoforms, nNOS. Don't worry about that, but just know that there is this enzyme, nitric oxide synthase is the enzyme, right? And it requires 5 cofactors. It requires 5 cofactors. Yes, HNN. It's better always. All of you have such pretty and good names. Why don't you keep your names as your ID so that you also feel proud of your name, right? 5 cofactors are NADPH, FAD, FMN, heme, and oxygen, right? So there is a transport of electron that is going on here. What is going on? Transport of electrons. So first electron is transported to this, then this, like the electron transport chain. Similar kind of thing is going on. And there is one more entity. Tetrahydrobiopterin is also present there to support the electron transfer things, right? So these are the 5 cofactors, right? NOS is the enzyme that is involved, right? Let's talk about the other ones. Tyrosine. Tyrosine gives you what? Tyrosine gives you what? If you metabolize the tyrosine, that gives you what? Melanin, your skin pigment, your thyroid hormones, right? What else? Catecholamines, dopamine, epinephrine, norepinephrine, right? These all are the metabolic products of the tyrosine. And one point, don't forget here, tyrosine, which we have synthesized from the phenylalanine. So we can say these are the products of the phenylalanine as well. They may trick you via this, right? Histidine. Histidine. I'm writing on the top histidine. Histidine gives you what? Histamine. Histidine gives you what? Histamine, right? And what kind of a reaction is that? That is another important point for the examination point of view. What kind of a reaction is that? That is a sort of a decarboxylation reaction. That is a decarboxylation that is going on. You all are well prepared. I'm really proud of you. Really feeling proud of you guys. Okay, let's move forward. Question number 4. Most abundant amino acid that is found in collagen. Very, very hot topic nowadays. Collagen, 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 collagen. Okay, what is most abundantly found in collagen? Glycine, proline, hydroxyproline, lysine. Glycine is most abundantly found in the collagen, right? Glycine is most abundantly found in the collagen. For those who take my class, they are very familiar with this picture, right? This is the collagen fibril. No, not hydroxyproline, Ayushi. Glycine is the one, right? So this is the triple helix, right? There are so many triple helix, and in which we bundled together to make the collagen. So triple helix, as the name is suggesting, is composed of 3 units. If you take 1 unit out, that is a right-handed alpha helix, right? You take 3 right-handed alpha helix and then you twist into the left-handed form. That makes 1 alpha helix. So now we are separating all the helices of this alpha helix and we get 1 alpha chain. And what do we see? What kind of a pattern we are seeing? Gly-XY, Gly-XY, or we can say X and Y are your what? Your proline and the lysine. So every third residue is what? If you take a clearly look at it, what is this? Every third residue is glycine is the for sure. So glycine is the most abundantly found in the collagen, not the proline, not the hydroxyproline, not the lysine, because we are sure that the third one is the glycine only, right? Afterwards, the lysine and the proline, they get hydroxylated, they get glycosylated, right? And then we bundle them together, right? Other important point, where do we see the glycine? Where else we see the glycine? In the heme synthesis. Remember, in the heme synthesis, glycine was there. Purine rings. When we synthesize the de novo synthesis, purine has two ways to synthesize, de novo and the salvage pathway. During the de novo synthesis, right, we used glycine, but not in pyrimidine. Make a note of it. Don't make fool of yourself, right? Only the purine. Who else? Glutathione formation. Glutathione is what? Glutathione is a tripeptide. What is glutathione? Glutathione is a tripeptide. Tripeptide means it has 3 amino acid, you can say. One is gluta. First word is gluta. Gluta comes from word glutamate. So one amino acid is glutamate. The second half is thio. Thio is coming from cysteine, right? And one. One means last thing. If you look at one, very first amino acid is glycine. Simplest amino acid is glycine, right? And then bile acid conjugation also we have seen the glycine, right? Your cholic acid, chenodeoxycholic acid, and it also acts as neurotransmitter. So glycine is very important amino acid, though it is very, very simple in structure. Which of the following is considered as 21st amino acid? Since till our high school we knew about 20 amino acids. We cannot say which one is the number 1 amino acid, number 2 amino acid, number 3 amino acid. We cannot number it. But yes, with the time we figured out there is 21st amino acid, right? So we called it 21st amino acid, and that amino acid is selenocysteine, right? Is the 21st amino acid. That is the choice B. Pyrrole lysine is your 22nd amino acid. 22nd amino acid. Very good. Very good, all of you. I'm really proud of you that you are getting it right. Right? So pyrrolysine is 22nd amino acid, and we should know few very important facts about the selenocysteine. Right? Selenocysteine, as the name is suggesting, right? See, seleno and cysteine. Cysteine contains the sulfur, we know that. If we take a look at the structure of the amino acid, you don't have to memorize, but it always gives you a good insight. This is your serine. This is what? This is your serine. From serine, we can synthesize the selenocysteine. First of all, remember this. From where we synthesize the selenocysteine? We synthesize the selenocysteine from serine. What we have here? Selenium. What kind of here we don't have nitrogen, we don't have oxygen, but we have selenium. So from serine we synthesize selenocysteine, we write it in the form of the sec. But if we take a look at the structure of the cysteine, it is similar to the selenocysteine, but it has what? It has sulfur in the place of the selenium, right? Or we can say they both differ by the selenium and the sulfur. So we can call selenocysteine as the selenium containing cysteine, right? Hence the name selenocysteine. But don't get confused, selenocysteine is synthesized from the serine, not from the cysteine, right? And what is the codon that codes for the selenocysteine? UGA. What do we know about UGA? UGA is a stop codon. UGA is a stop codon. So it is synthesized when the selenocysteine is synthesized during co-translational mechanism. Okay, it is polar, it is acidic, and it is a strong nucleophile than cysteine, and it is a very good antioxidant. And from where we can get the selenocysteine? From the animal sources, like for the people who eat meat. From the plant we get it from the wheat, oats, corn, Brazil nuts. And there are few other important facts we should be knowing about the selenocysteine, right? The Keshan's disease, right? Deficiency of selenocysteine leads to the Keshan's disease. Another important question for the examination point of view. It has a cancer prevention effect. It helps in the thyroid hormone regulation, and its deficiency leads to the lean body mass, prone to premature aging and the heart diseases, right? Another important thing here, HIV encodes of functional selenoprotein. Patient with HIV, right, have decreased plasma selenium. That is how the HIV virus survives. Or in another way, we can say selenium gives you a protective covering against the viral infection. That is why in the COVID era, since we are in the COVID era, selenocysteine is good for health. So we should be eating the selenium containing compounds. That is like the mushrooms, the rind of the watermelon, right? Deficiency increases the risk of the viral infection. So know these point number 5 and 6 as well, right? And these are the enzymes which contain selenocysteine amino acid, glutathione peroxidase, triiodothyronine deiodinase, thyroredoxin reductase. What is the common thing here? Thio. In the class, in the chapter, I said when you hear the word thio, that is coming from sulfur. So imagine that is a sulfur containing thing. But also in the selenium containing amino acid, the selenocysteine is also with the name thio. So remember, these three are important enzyme we should be knowing for the examination point of view. Okay. Question number 6. A child presents with complaints of urine turning black on standing. What is the diagnosis? Urine turning black on standing. What is this? This is Alkaptonuria. We know that for sure, right? This is the Alkaptonuria, right? This is the pathway of the degradation of phenylalanine. Phenylalanine gets converted into tyrosine, and which is the enzyme that is involved? Phenylalanine hydroxylase or phenyl hydroxylase or phenylalanine hydroxylase. In the short form, we call it as PAH, right? And the gene that encodes the PAH is PAH gene. If there is that defect in the gene, that leads to the phenylketonuria. That leads to what? Phenylketonuria, right? But here we are talking about the urine turning black upon standing, and that is what? When tyrosine is getting converted into 4-hydroxyphenylpyruvate, which will get converted into homogentisic acid. This homogentisic acid should be degraded to form methyl acetoacetate, right? Don't worry about the name of the substrate, just figure this out. Homogentisic acid should be converted via the enzyme homogentisate oxidase or homogentisate dioxygenase. Oxidase or dioxygenase, both are the same things, right? Just don't get confused with the names. They may call it oxidase, that is an old name, but now in today era we know this is a dioxygenase, so they may give you the dioxygenase, but many a times they trick you by writing dehydrogenase, right? So don't fall in that trick. That is an incorrect option. Homogentisate dehydrogenase is no, not a good choice. Dioxygenase or the oxygenase, right? If this enzyme is deficient, that leads to Alkaptonuria. That is AKU, on which we say always the urine turns black upon standing, right? If you have the urine in the tub, you leave it for a particular point of a time, the urine will turn black upon standing, right? And this is the picture I always give you, and I will keep on giving you the same picture over and over so that you get hang of it, how to read from the book. That is always a question, how to read from the book, how to read from a book. If I'll keep a writing by my hand, how you're going to get hang of looking at the figures and the diagrams written in the book? Okay, so I'll keep doing it until you get used to it. This is an autosomal recessive, so homogentisic acid is not degraded, so there will be lots of homogentisic acid. They will start polymerizing each other. Initially we will not know it, right? When we are baby, infant, like we will become teenagers, we will not know it. Eventually with the time, pigmentation will be increasing, like the deposition quantity will be more and more and more, then what we will see, the blackening here in the ear, in the eye, right? And when the testing will be done, then the urine will turn black on standing. It was there since the childhood, right? It was there, but usually what, after urinating we flush, so we don't notice it until unless it appears physically on the surface. We can see it by the eyes only, right? Even if we keep the diaper of a newborn baby and that is suffering from the Alkaptonuria, if you keep the diaper, then the diaper will become black because the urine will turn black upon leaving, right? So it is initially asymptomatic. Symptoms are seen with the age, and patients in the old age, let's say 30s, late 30s, early 40s, they may experience the stiff back pain, like in a lower back pain here, like an arthritic kind of a pain. They may feel that stiffness also. Okay. And this is the mnemonic I always give you. Alka came home and saw black bug, shouted, "Oh, Alka, Alkaptonuria, homogentisic acid, urine turns black on standing," and ochronosis, definitely with the lower back pain or the stiffness. Question number 7 is, a 40-year-old man presented with black pigmentation of the pinna, sclera of the eyes, urine of color changes to black on lone standing. See, they have given you the questions again. They are repeating the question. They are just rephrasing the same question over. We know this is Alkaptonuria. Question number 8 is, which structure of protein is not denatured? Not denatured. So we know this protein is the primary, secondary, tertiary, and the quaternary structure. In the quaternary structure, you have one peptide, two peptide, maybe 3 or 4, they are staying together. If you heat them up, they are going to get separated, because what they have kind of an attraction towards each other, and that's it. So you can imagine you are 4 friends sitting right in a room. As soon as the heating, mom come heat it up. Okay, what you are doing, why you are wasting your time? You are gonna separate. Same thing, all peptides will be separated. So quaternary will be gone. Tertiary structure is the same way. They have what kind of interaction? They have mostly the non-covalent interaction, van der Waals, electrostatic force of interaction, hydrogen bondings between the amino acids, right? So even they will fall apart when heated. Secondary structure, alpha helix, beta sheets, right? Again, what they are? They are same kind of interactions. This will go. Only the primary, in which the backbone is really bonded to each other by the covalent bonds, right? A fevi-bonding kind of a bonding, that is not gonna go away until you heat up beyond 700 degrees Celsius, right? Beyond 100 degrees, okay? So we will choose A as the answer. Yes, perfect. Yes, you all are doing very good for exam. Question 9, ammonia formed in the brain is converted into, come on, ammonia formed in the brain is converted into what? This is the ammonium ion. How we get rid of the ammonium ions? It is the same, whether it is a brain or in the liver. It is the same way we get rid of the ammonium ions. That is in the form of the glutamine, right? And this is a picture I have gotten for you from a very good journal, right? You should be knowing where it is happening. This is happening in the astrocytes. They save your brain cells, right? So what do we see here? See, look at here. This is alpha ketoglutarate is taking up the ammonium ions, getting converted into what? Glutamate. And the enzyme is GDH, glutamate dehydrogenase, right? Now you have the glutamate, then this glutamate can take up one more ammonium ions, right, and can get converted into glutamine, and the enzyme involved is glutamine synthase, and that glutamine can take these two ammonium ions, these two nitrogen, right, ammonium ions out of the astrocytes. That's how you do it. Since you have the choice of the glutamine, you may see the glutamate, pick any of them. But what if they don't give you the glutamate or the glutamine? See here, the third half, hidden portion. But you have, see, when you formed the glutamate, the first part, when it formed the glutamate, right? The glutamate can give the ammonium ions to whom? The pyruvate, the upper half, and get converted into the alanine. So another one is the alanine, if they are gonna really trick you bad. Okay, so first choice is glutamine, then you have the glutamate, and the third choice is the alanine. If you have all three, pick the glutamine. If you have none, pick the alanine. Question number 10. Nitrogenous waste are excreted from the body in the form of. Earlier the question was from the brain. Now they are asking you question from the body in the form of what? Glutamine. Do we secrete glutamine? Do we secrete urea, uric acid, or nitric oxide? Come on. We secrete urea. Many times many students, they get confused between urea and uric acid. Uric acid is the product of the purine pyrimidine metabolism that we see in the nucleic acid chapter, right? It is not the form we excrete nitrogen out of the body, right? So don't get confused between urea and uric acid. They are different entities. Glutamine is the way we transfer ammonium ions from the peripheral tissues to the liver. Okay? Finally, liver will be the place where urea cycle is going to take place. But you have ammonium ions all across the body, right? So glutamine is sort of a vehicle, or you can say a porter, which transports ammonium ions from the brain, such as from the brain to the liver. Okay? 11 is melanin is derived from which amino acid? We have just did it. Melanin is derived from where? Melanin is derived from? Come on, give me the answer. Melanin is derived from tyrosine. Many a times we get confused because there is another term, melatonin. Melatonin is not tyrosine, but melatonin is tryptophan. There is another thing, serotonin. What is serotonin? Also known as 5-hydroxytryptamine. Vitamin B3. These all three are the products of the tryptophan, and how we are going to memorize it? Tryptophan is 2-2-2. So we will be 2-2-2, right? Trypto, melato, seroto. Okay, so these are the product of trypto, not the tyrosine. Tyrosine is giving you what? Tyrosine is gonna give you the melanin, T3, T4, catecholamines, dopamine, epinephrine, norepinephrine, all those things. Okay, perfect. Let's move. Okay, so here melanin again. Right, tryptophan is serotonin, totonin, and vitamin B3. Right, glycine is, I have told you already, the heme, purine ring, glutathione, creatine, histine, you can synthesize histamine, arginine give you nitric oxide, which is a vasodilator. Perfect. Question number 12. Vitamin K2 is known as, we are talking about vitamin K, that is a fat soluble vitamin. That is a fat soluble vitamin, right? And K is coming from coagulation. So this vitamin is participating in the coagulation. It has three forms: vitamin K1, K2, K3. So calcitriol is D3. So we can cross this option out. Now we are left with the A, B, C. All three are vitamin K, right? Phylloplant is K1, menaquinone is K2, menadione is K3, right? So this is the only fat soluble vitamin with the specific coenzyme activity. Again, remember this for examination point of view, right? So K1, K2, K3, and how we are going to remember it? Phyllocunone, right? Second one will always be, another again will be quinone, that will be menaquinone, and then third will be dione, right? The spelling gets shorter. Remember this way, like stupid way, but this is the way I have learned it. So I can tell you this much only, right? 13. Question number 13 is biotin acts as a cofactor for which of the enzyme? Biotin is cofactor for which of the enzyme? All the carboxylases, mainly, like there are exceptions, but for the examination point of view, carboxylases, they contain a biotin. What else we should be knowing about the biotin? Biotin is vitamin B7. Earlier it was called as vitamin H also. So they are the other names of the vitamin, right? And it is the cofactor for the hydrocarboxylases enzymes. ACC, acetyl coenzyme A carboxylase. Where we have seen it? We have seen it in the fatty acid synthesis. Where we have seen it in the fatty acid synthesis. Pyruvate carboxylase. Our PC, gluconeogenesis. Propionyl coenzyme A carboxylase for the odd chain fatty acid synthesis. Methyl crotonyl coenzyme A carboxylase. Right? What is the common point between all these things? They are what? They are all carboxylases. So biotin is required by all the carboxylases, and the deficiency is rare, but there are one of my very good friend, he wanted to lose the weight and he started eating the raw eggs. Raw eggs has what? Raw eggs has a avidin, right? Raw egg has a protein avidin, which interferes or which dilutes or which degrades the biotin. It does not allow biotin to stay in the system. So it is leading to the biotinidase deficiency, and that is really, let me tell you guys, very lethal, very dangerous. So like if you are eating the raw eggs for the prolonged period of time, that is going to decrease your biotin level down, and then you are going to see that person in the ER. Okay, symptoms of the biotin deficiency is the alopecia, bowel inflammation and the muscle pain. But remember, usually it is a rare disorder, right? But if the raw eggs are eaten for the prolonged period of time, definitely it is going to show its effect. Now question number 14, which vitamin deficiency causes burning foot syndrome? Riboflavin does what? Riboflavin is gives you what? Riboflavin is giving your glossitis, chiliosis, right? And corneal vascularization. Yes, choice C is the right, pantothenic acid, right? So vitamin B2 deficiency is glossitis here, and the corneal vascularization. Niacin is related to the pellagra, right? Your D3s or D4s, you can say diarrhea, dementia, dermatitis, finally maybe leading to the death. Thiamine gives you all sort of beriberi, your wet beriberi, your dry beriberi, infantile beriberi, Wernicke-Korsakoff syndromes. All these are because of the thiamine deficiency, right? Wow, very good. All of you are doing great. Ariful, Ayushi, Wasim, Ashish, Robin, Anil, Gowru. Right, perfectly, perfect. Okay, so vitamin B5 is the one which is leading to the, right, and where we need it, it is in part of a process where we are synthesizing the acyl coenzyme A, right, during the fatty acid biosynthesis, right, because we have to synthesize coenzyme A. So whenever you have to synthesize coenzyme A, you will be needing vitamin B5, right? And where do we see the acyl coenzyme A? In the citric acid cycle, fatty acid oxidation, acetylation, cholesterol synthesis. These are the part where you see the coenzyme A taking part, is like participating. You should know this also, right? This is another way to look at this question. And the enzymes that is directly using B5 as a cofactor is fatty acid synthase, fatty acyl coenzyme A synthetase, pyruvate dehydrogenase, alpha ketoglutarate dehydrogenase, right? And the dietary deficiency can cause peripheral nerve damage, but still it's rare, right? Right? So, and this is from the notes also, it is Gopalan burning foot syndrome, right? And but for FMG, very important. Question number 15th. Yes, Satvik, I am teaching, but sure you will see me again. Thank you for encouragement. You will definitely see me soon. Which enzyme level is tested in thiamine deficiency? What is thiamine deficiency? Vitamin B1 deficiency. Okay, where we see the thiamine, which enzyme is using the thiamine? Okay, come on. Which enzyme needs the thiamine? If they don't have the thiamine, they are going to die, right? And if we check them, they are working or not working, we can definitely test whether the thiamine is present or not, right? So for that, we should be knowing which enzyme needs the thiamine. pyruvate dehydrogenase, in the Krebs cycle alpha ketoglutarate dehydrogenase, in the HMP shunt the transketolase, or I call it as a HTK, right? And your BCKTH enzyme, branch chain keto acid dehydrogenase enzyme. One very another important point of here, this is the maple syrup. This is your maple syrup urine disorder. Urine disorder, right? Because this there is this sweety fruity smell of the urine. Nobody wants to smell the urine, but there is a burnt sugar, or you can say the caramelized, right? When we cook, sometimes the sugar gets burnt, that kind of a smell is there. So that is because of this enzyme deficiency that needs the thiamine also, right? And also for the acetylcholine formation, it regulates the chloride channels in the nerve conduction, CFTR we know, right, for the cystic fibrosis. So what and which enzyme we see? We see PHD, they should have written PDH. That is a transketolane enzyme level is tested in the thiamine deficiency because it requires thiamine as a coenzyme. What is the coenzyme form? It is TPP. It is not the vitamin B1. It is thiamine pyrophosphate is the coenzyme form. We should be knowing the coenzyme form. Okay, let me ask you, what is the coenzyme form of vitamin B6? Anybody? What is the coenzyme form of vitamin B6? Come on, anybody? That is PLP, pyridoxal phosphate, right? This is the coenzyme form. Question number 16, most sensitive screening test for vitamin A deficiency. Very easy question. We don't have to think anything about it. That is why whenever we think about vitamin A, we are talking about the eyesight. And since childhood, mama always keep on saying, eat carrot, eat carrot, eat carrot, eat carrot. Carrot. That is beta carotene, right? So vitamin A, if it's deficient, it's a problem. If you have too much of vitamin A, again a problem. So deficiency system, earliest sign is, first of all, sensitivity of the green light will be gone, right? Then followed by the impairment of the adaptation of the dim light, right? If you are in the bright light and if you will come into the dark room, you will be having a hard time, person will be having a hard time to see. And then finally, the prolonged deficiency of vitamin A will lead to the keratinization of the cornea and the skin and the night blindness, okay? What else we see? Xerothelmia and the Bitot spot. Thank you, Meghna Sharma, thank you. Fifth is follicular hyperkeratosis, alopecia we see, right? And vitamin A deficiency leads to gonadal dysfunction in males and miscarriage in the females. A leads to toxicity also, right? Hypervitaminosis, right? Because of self-medication, and this era, most of the people, they think they know a lot, they don't go to see the physician, they don't come to you, they'll rather start doing the self-medication, right? And you know, eating liver of the seal and the polar bear can lead to hypervitaminosis. Why? Because 30 gram of polar bear liver contains 450,000 IU, and if you have too much of the vitamin A, what will happen? Continued ingestion of the vitamin A will lead to the peeling of the skin from the head to the foot, okay? Next question is, patient present with drying of skin and appears, this is the picture we have been given, and whenever I look at this picture, what do I think of? Biotin deficiency, niacin deficiency, riboflavin or ascorbic acid? This is nothing other than the niacin deficiency, right? Vitamin B3 deficiency it is, right? And vitamin B3 niacin is synthesized from the amino acid tryptophan. This is there we have atypical vitamins, right? Niacin is one of the atypical vitamin, another is the vitamin K, right? So tryptophan, from tryptophan, 60 milligram of tryptophan gives you 1 milligram of the niacin in the body. Okay, and the functional form of niacins are NAD and NADPH, and they are required, we know that they are required everywhere in the body. Names of vitamin B3, we should be knowing niacin, nicotinamide, vitamin P, because deficiency is going to give rise to the pellagra-like conditions. Anti-black tongue factor, nicotinic acid, these are all the names of the vitamin B3, right? And also what we see in relation to the niacin deficiency, Cazal's necklace, any kind of the rashes around the neck region, right? And that leads to the 4 Ds, diarrhea, dementia, dermatitis, and maybe finally to the unfortunate death. Very good, Ayushi, very good, right? And the people who are on the prolonged diet, they keep on eating the maize or the sorghum, that is hampering the synthesis of the niacin or maybe the absorption of the niacin. So those who are for the prolonged time on the maize or the sorghum diet, they are going to be suffering from the pellagra-like condition. Pellagra can be 2 types, primary pellagra, secondary pellagra, right? If we don't take the diet which has the niacin or the tryptophan, that is a primary pellagra. Second thing is, you are taking it in the diet, you have very good diet of tryptophan, everything is well, but our system is not able to absorb it properly, that is secondary pellagra. But this is not important. What is important, the pellagra-like symptoms can be seen in 3 cases. First is just a simple niacin deficiency. Second is pellagra occurs in the heart nerves disease. Third is it also occurs with the carcinoid syndrome. So in the exam, you should be able to distinguish whether the examiner is talking about just the pellagra, that the niacin deficiency, or the heart nerves disease or the carcinoid syndrome. How we are going to do that? Heart nerves syndrome, there will be what? The excess renal loss of the niacin. In one way or another way, they are going to give you this hint in the scenario, clinical case scenario. In the carcinoid syndrome, there will be increased synthesis of the 5-hydroxyindole acetic acid, right? So all the tryptophan is taken toward that pathway. From the tryptophan, lots of serotonin is formed, and the serotonin will be converted into 5-HIAA, and none is available for the other functioning. That is why the patient will be showing the pellagra-like conditions. Okay, so remember this for examination point of view. Question number 18, which of the following is one of the uses of dietary fibers? Okay, they are asking you about the uses of the dietary fibers. What they do, they reduces the intestinal transit time, right? They help maintain blood glucose level, they boost up the metabolism, right? They prevent constipation, right? They reduce the risk of the heart disease, diabetes, constipation issues, right? And the soluble dietary fibers are found, we can get from the oats, flax seeds, beans, oat bran. That is why they are prescribed for the patients who are suffering from the high cholesterol, because they help reduce the blood cholesterol levels. Okay, and it is speculated to be why this good things are happening because of the soluble dietary fibers, because they change the intestinal viscosity, they help in the nutrient absorption, the entire food is passing from the intestine, the production of the short chain fatty acids which are good for us, and production of the gut hormones which helps us to digest and improve our metabolic functions. Question number 19, 1 gram of protein yields how much of energy, right? Carb, we eat lipids or triglycerides, triacylglycerols, whatever you want to say it, protein and alcohol. We should know about all these 4. Question is asking you about protein. Protein is how much? 4. They may write cal or they may write kilocal, one and the same thing. Same number is for the carb, right? So carb and the protein is 4 kilocal, right? Alcohol is 7 kilocal. Yes, my dear Yes, my dear, alcohol gives you the calories. It doesn't mean you keep on drinking or somebody keeps on drinking the alcohol all the time. Yes, but it has a caloric value to it. Lipid gives you the most, or triacylglycerol gives you the most, that is 9. So answer to this is the choice C. Yes, yes, Pranav. Yes, Mullai. Yes. Right, Keshan's disease. I have just told you in the previous question, Keshan's disease is related to the deficiency of what? Selenium, zinc, copper, iron. Iron will give you anemia. Where else we see anemia? With the folic acid, right? Vitamin B12 also gives you anemia, different kind of anemia. Copper deficiency is related to what? Menkes disease. Right, selenium, perfectly fine. Zinc is related to what? Wilson's disease. Zinc and copper go hand in hand. Zinc helps in the absorption of the copper, right? If copper level goes down, zinc level goes down, copper levels are elevated. That what is happening in the Wilson's disease, okay? So zinc helps in the absorption of the copper. Very good all of you. Which of the following is not a micronutrient? Zinc, copper, calcium, iodine. Again, a very straightforward. Calcium we need in lots of amount as compared to rest. Yes, Abhishek, acrodermatitis enteropathica. Very good, Abhishek. Zinc is a cofactor in which enzyme? In carbonic anhydrase. Lysyl oxidase contains what? We have studied this in the collagen synthesis. Lysyl oxidase is the enzyme to use for the collagen synthesis. That uses what? Copper. Carboxylase uses biotin. What about deiodinase? Deiodinase uses what? Deiodinases has FMN. Deiodinase is FMN as a cofactor, okay? Deiodinase is used for controlling your thyroid hormones level. Question number 23. Which of the following options given below explains non-competitive inhibition? Okay, there is no inhibition at all. What it is gonna do? We know inhibition can be of two types, majorly competitive inhibition, non-competitive inhibition, right? And both kind falls under the category of the reversible kind of inhibition. There is another irreversible, that is suicide kind of inhibition. But reversible inhibition is competitive and non-competitive. And we can distinguish between based upon the values of the Vmax and the Km. Vmax and the Km, right? In competitive inhibition, Vmax remains the same. In non-competitive inhibition, Km remain the same, opposite. In competitive inhibition, Km increases. In non-competitive, Vmax decreases. We have to learn this way. What the hell I am talking about? So dear friends, so when we have this kind of a chart, let's say the x-axis is the substrate concentration and this is the rate of reaction. That is V. Initially, there are enzymes, there are substrate, they are going to come each other, they are going to like be a lovey-dovey to give you the products, right? And the rate of reaction will keep on increasing until 100% of the enzymes are saturated with the substrate and they are making the products. So the rate of the reaction will reach up to a level and it will level off, because all the enzymes at this point in time is occupied. So this is known as Vmax. This is the highest velocity you can get. If you make the half of the Vmax, this is the point when the velocity is at half of its value, and you drop it down on the x-axis, you will get the substrate concentration when Vmax is half. This substrate concentration is your Michaelis constant. Michaelis constant is nothing but the substrate concentration when velocity is half of its maximum volume, right? So in the competitive and non-competitive inhibition, we play around whether the Vmax is the same or Km changed or not. So remember, competitive, non-competitive, they work in the opposite way. In competitive, Vmax is the same In non-competitive, Km is the same, right? And competitive, Km increases, non-competitive, they don't have any competition, so the velocity decreases. Just remember this way, okay? If you can remember this from the class also, they may give you the graph also. So here is the direct, that is Michaelis plot. This is the double reciprocal plot for them, right? So this is a competitive inhibition. What we know about the competitive inhibition, this Vmax remains the same and the Km is changing. This purple color is with the inhibitor. Km changes. But if you take a look at the double reciprocal plot, what we know, Vmax remains the same. Vmax is your y-intercept right here. That is 1 over Vmax. So the competitive will be like the scissor. This is competitive will be like the scissor. So remember this. This kind of a graph means the competitive inhibition, because the point where they are intersecting, that is when Vmax is the same, but see Km is changing. For the non-competitive inhibition, they will have a same start point. I'm telling you a trick to remember the graph. They will have a same start point. Since they are not competing for a girlfriend, they will have two different ways. So Km is the same, but the Vmax right here, the y-intercept is different. It changes, okay? And here they are parallel, because they don't have the same girlfriend to fight on. There is no love triangle sort of a thing. That is non-competitive fighting. Uncompetitive fighting, similar to the non-competitive fighting, see they're both graphs here and here, almost kind of a same, but double reciprocal is now parallel. Km is also different, Vmax is also different. That is a rare case you can get. Most of the times you see in the exam is competitive and the non-competitive. Competitive, tug of the war, like ship, right? And this non-competitive, they are going their way. They are finding their different ways. It is like a victory. 24 question. Dash is not derived from cholesterol. Vitamin D, skin, that is a cholesterol, right? That is a cholesterol. Bile salts, that is derived from the cholesterol. Bile pigments are not derived from the cholesterol. Steroid hormones all are derived from the cholesterol. So very straightforward answer, that is choice C. Bile pigments are not derived from the cholesterol. Acetyl coenzyme A can be converted into all of the options given below except acetyl coenzyme A. So we are talking about acetyl coenzyme A. Can we synthesize? Can acetyl coenzyme A be glucogenic? No. Right. Ariful, Govra, very right. Acetyl coenzyme A synthesize fatty acids. We need it for the cholesterol biosynthesis, and we also need it for the ketone bodies synthesis. So the choice is A. Number of ATP produced in palmitic acid oxidation. Palmitic acid oxidation, whenever we talk about what the beta oxidation, in the class also the beta oxidation we talked about is the one we always say ki palmitic acid is the one we are going to take, right? That is 106. You have glycolysis, you have citric acid cycle, you have beta oxidation. All these pathways gives you what? Gives you ATP. So if you have glycolysis, if we have glycolysis can be aerobic, anaerobic. How much ATP is in the aerobic? How much ATP is in the anaerobic? Aerobic you get 7 ATP. Remember this, take these notes down for the examination point of view. Okay, anaerobic is 2 ATP. Then you have citric acid cycle. Citric acid cycle gives you how many? 10 ATPs, right? And when you talk about the palmitic acid oxidation, which is a by default we take for the beta oxidation, that is 106 ATPs. Perfectly found. This is from the class, right? So this is your palmitic acid, right? And from palmitic acid, if we take out one acetyl coenzyme A, it releases one FADH2, one NADH, right? When we have to chop it off, it gives you one NADH and one FADH2. If we repeat it for the certain interval of time, Palmitic acid is C16, right? If you chop it into pieces of 2, 2, 2, 2, 2, how many acetyl coenzyme A you are going to get? You are going to get 8 acetyl coenzyme A. But the last step, there will be piece of 4, and when you will chop it off, there will be 2 and 2, right? That will give you 2 acetyl coenzyme A. So the number of cycles it will go through will be 7 only, right? So you will be having 7 NADH, 7 FADH2, 1 NADH is 2.5 ATP, 1 FADH2 is 1.5 ATP, and again you can do the maths. I'm going to save time because I have lots of other questions to finish off, okay? Which of the following is cardioprotective, right? Cardioprotective, we are talking in the terms of we don't want too much of the triglycerides. So when we talk about the lipoproteins, the chylomicrons, your LDL, HDL, IDL, all kind of things, which one has the least amount of triacylglycerols? This is from the class again. You have the chylomicrons, VLDL, LDL, and the HDL. HDL is the one, right, which has the least of it. So this is a cardioprotective in a way, we can say. Which among the following is most essential amino, not amino acid. This should be fatty acid. I'm sorry, that's a typo. Most essential fatty acid, linoleic, palmitic, arachidonic acid. This is definitely not an essential amino acid. Oh, sorry, fatty acid. So linoleic, linolenic, arachidonic. All these are essential fatty acids. Which one is the most? Which one is the most? A, B, C. A is the most essential amino acid among linoleic, linolenic, arachidonic, right? If we go back from the class when we classified the lipids, one classification was based upon the synthesis. Essential fatty acids, non-essential fatty acids, right? Essential fatty acids which cannot be synthesized in the body, right? We have omega-3 and omega-6 fatty acids. Linolenic is omega-3 fatty acids. Linoleic is omega-6 fatty acid, right? But we have a gamma-linolenic acid and arachidonic acid as well. So this is the one which is most essential if we have to talk about the essentiality. Right? It is like when you are dying, which one you want the most, water or the air? You will say air, right? Definitely we will be needing a water if we are very, very thirsty. But it is like linoleic is the one. Right? And linoleic we can get from the safflower oils, contains the most amount of the linoleic acid. It is also found in the corn oil, sunflower oil, soybean oil, right? And for arachidonic is coming from mostly the animal sources. DHA is coming from the breast milk, okay? Nucleotide. Nucleotide. We know two terms, nucleoside, nucleotide. Nucleoside is, there are three entities in there, base plus sugar. Base plus sugar is a nucleoside. But if you attach with the sugar, if you throw in the one phosphate, that gives you nucleotide. And how we remember it? How we remember it? My marker is not working well. This is T. This is T. Remember this, and this should have a T. Phosphate has a T. So remember, so nucleotide has a phosphate with the nucleoside, because nucleoside is base plus the sugar. So choice is C. Very good all of you. Question number 30. Post-transcription modification takes place at which kind of a post-transcription modification takes place at the 5' end of the RNA. So whenever RNA is synthesized, what we have heteronuclear RNA. Right, and that should go under post-transcriptional modifications, and there are three kind of post-transcriptional modifications we should be aware of. One is at 5' end, one happens at the 3' end, and one is happening with the introns. Right, introns are the intruders which should be removed from the RNA. At the 5' end, what we add 5' methylguanosine cap. We added, this is like a baby is born. As soon as the baby is born, we put a cap we put a shoes, that is a tail, right? And then we wrap it up by removing the extra things, that is removing the introns via the spliceosomes, right? They are also known as snRPs. SNRPs does the spliceosoming. So 5' methyl guanosine, 7 methyl guanosine in the 5' end, that is a cap, and at 3' end we add what? A poly A tail. Lots of A's are added to preserve it. So it is 7 methyl guanosine, that is choice C. The bases which are present in human DNA. Do we have uracil? No. Do we have tyrosine? No. And also remember, whenever we have adenosine, cytidine, thymidine, uridine, these are what? These are nucleoside. This is not a nucleotide we are talking about, right? And in DNA what we have? We have a nucleotide. So we should be talking about AMP, GMP, CMP, TMP, UMP. UMP is definitely in the RNA, not even in the DNA. So they may make a different versions of the same question. So know about this, okay? Reverse transcriptase is used in what? Reverse transcriptase. So we know DNA, central dogma, DNA to RNA is a transcription, right? Is transcription. So RNA to DNA is a reverse transcription, right? So enzyme is a reverse transcriptase. So reverse transcriptase is we are doing what? Are we amplifying the RNA? Yes, we are amplifying the RNA. We are not amplifying the DNA. DNA dependent DNA polymerase, for that we need a DNA as a template, but we are using RNA as a template. RNA polymerase will do the transcription. DNA amplification is again kind of an transcription process which we use in the PCR. RNA amplification we do in the RT-PCR, right? That's how we are amplifying the COVID virus, right? RT-PCR, that is example of an RNA amplification. DNA dependent DLA polymerase, what we are talking about here? We are talking about the replication. DNA dependent DNA polymerase is a replication process. Ayushi, it's A, RNA amplification, okay? Question number 33. In a given DNA, we are given A is 15%. So A is 15. So A is going to combine with T, it will be 15. So what will be? Total is 30, right? So out of 100, 30 is A and T, left is 70. We know G combines with C, so they are half of it. So we can say 2G is 70, that is 35. Is that making sense? Whatever, how many A's are there, there should be an equal number of the T. So A is 15, so T is 15. So combined is 30. Keep them apart. What we are left with? We are left with 70. That should be distributed equally between G and C. So 70 divided by 2 is 35. As simple as that. This is nothing but this is a Chargaff's rule. Perfect. Yes, Pranav. Yes, Ariful. Ayushi. Perfect. Which is the specific test for uric acid for the gout? How do we measure the uric acid levels in the gout? We measure the synovial uric acid. Why synovial uric acid? We know the purines degradation are gonna produce the uric acid, right? And these uric acids are going to deposit in the form of what? In the form of what? Crystals. They make what? Urate crystals. They make urate crystals. They make urate crystals which accumulates where? In the, yes, in the joints, right? And what kind of urate crystals are those? Those are monosodium urate crystals, right? And what are the shape, Ayushi? What is the shape? Needle shaped. They are needle shaped. So we have to check since they are depositing in the joints, right? The toffee here, the big toe we always talk about it for the gout, right? So we have to check the synovial uric acid levels to test for the formation of the crystals. Next question is, the disease Lesch-Nyhan syndrome occurs because of which deficiency? That is hypoxanthine guanine PRTase, HGPRTase is Lesch-Nyhan syndrome. And what do we know about the Lesch-Nyhan syndrome? Self-mutilation. The patient is gonna bite itself. It will bite the lips. It will chew the fingers, not just biting the nails, but the fingers. See, biting the lips. So we have to keep them on the pacifiers. They are kept held like they are tied with the ropes so that they don't eat their fingers, right? Definitely they will be having the gout, like uric acid level increased in the body, so they will be suffering from the gout and also the kidney stones. Okay, now here I'll guide you step by step, so just be with me. There are many things in this figure. So this is the purine degradation. Purine is pure as gold. So adenine and guanines are the purines. So here is your AMP, here is your GMP. They should be degraded, right? GMP is degraded, PI phosphate goes, it becomes guanosine, and then sugar goes, right? Then it becomes guanine. This is how GMP is degrading. Now important thing is about the AMP. When phosphate goes, it is nucleoside that is left, that is adenosine. Adenosine is deaminated to form inosine. The enzyme involved is adenosine deaminase. Enzyme involved is adenosine deaminase, also known as ADA, and it is related to SCID. This ADA deficiency is related to the severe combined immunodeficiency, right? That is ADA. This deficiency disease is SCID. One thing from this point, right? Then finally it will be converted into hypoxanthine. But when we know about the purine synthesis pathway, there are two ways to synthesize the purines. One is the de novo pathway, right, in which we also take help of the glycine. Another is the salvage pathway. Salvage pathway means we can recycle these two, right, that via the salvage pathway, and the enzyme involved is HGPRT. If that enzyme is deficient, that is going to give rise to the Lesch-Nyhan syndrome, and the characteristics are the spastic cerebral palsy, self-mutilation, hyperuricemia and gout, early death, and this is X-linked in nature, right? And when the uric acid level are increased, another important question for the examination, this has been previously asked in this, is allopurinol. Allopurinol is a prodrug, is used for the treating gout. Okay. So Lesch-Nyhan syndrome is X-linked recessive, right? You can have two cases. One is there is a complete deficiency of the HGPRT enzyme, right? Wasim, I'll hang on to this question. Just hold on to this question. If I cannot take it today because we have still 8 minutes left, tomorrow is another session at 6. That is a doubt solving question. I'll definitely address your question, Wasim. Okay. Another is the Kelly-Sieg-Miller syndrome, in which there is a partial deficiency of the HGPRTase. So remember this also, right? Kelly-Sieg-Miller syndrome, the enzyme is partially deficient, still it is working, right? And in those patients we see only the, we see only the gout. Okay. Complex 4 of ETC is inhibited by electron transport chain. We know in electron transport chain how many complexes we talk about: complex 1, complex 2, complex 3, complex 4, complex 5. Okay, very important question. You definitely gonna see a question from this electron transport chain, and you have to know this. They are not going to give you complex 1, 2, 3, 4 and 5. They will give you the name of the complexes, right? Complex 1 is NADH dehydrogenase. What is the name of the second one? I want you to tell me. What is the name of the second one? What is the name of the second complex? Anybody? Okay, answer is right, but I want the name of the complex 2. What is the name of the complex 2? That's what I want. Succinate dehydrogenase. Succinate dehydrogenase is the name of the complex 2, right? Then is cytochrome reductase, cytochrome oxidase, right, and F0, F1 particle. First thing one thing I want to make clear here, complex 1 is getting electrons from NADH. Complex 2 is getting from electrons from the FADH2, right? And this FADH2 is coming from where? Your citric acid cycle. Right. Your FADH2 is coming from the citric acid cycle. In the citric acid cycle, there are 4 oxidation reduction pathways. There are 4 enzyme dehydrogenases enzymes are taking place. One is succinate dehydrogenase. Right? So you can remember the name. You can correlate this name like that also. Right? One thing. Cytochrome oxidase is your complex 4. Right? CO, carbon monoxide. Carbon monoxide is a gas. What other gases are inhibitors of electron transport chain? Cyanide gas, hydrogen sulfide gas, right? So all these are going to inhibit the complex 4, right? So remember this one. Succinate, whenever we see the succinate dehydrogenase, it is inhibited by the malonate. It is inhibited by the malonate. Complex 1 is 1. Complex 1 is 1. Who is 1? Rotenone is 1. Phenobarbitone is 1. Right? So they are the inhibitors of the complex 1. Then you have the uncouplers. Uncouplers there are 2,4-DNP. Very, very important topic. Very, very important topic. 2,4-DNP is the uncoupler. Then you have the physiological uncoupler. Nowadays, physiological uncouplers are the examiner's favorite. Physiological uncouplers. Thermogenin. Ayushi, we should have one, but I'm not sure when. Thyroxine, right? Free fatty acids, right? They all are the physiological uncouplers. Know this. You are definitely gonna see a question in FMGE, and since Ayushi you are asking, remember this. This is a very important topic for the NEET PG as well. Okay. Which among the following transporter is insulin dependent? Insulin dependent transporters. When we talk about the GLUT transporter 1, 2, 3, 4. 4 is the one which is insulin dependent transporter. GLUT transporter, first of all, the mechanism they follow the ping pong mechanism. See, the hand is open. The hand is open to catch the ball, right? And then it is going to release the ball inside the cell. So that is ping pong mechanism is by the GLUT, right? GLUT 1, GLUT 2, GLUT 3, GLUT 4. GLUT 1, look at the GLUT 1 and GLUT 3. Km is 1, right? Km is 1. Km is 1 means Km is, if Km is low, that means they have what? They have high affinity. They have high affinity. High affinity, I always tell, affinity is love, affection, affinity, right? So they have high affinity for the glucose. So no matter how much glucose is there, they are gonna pick it up. GLUT 1 and GLUT 3. What is beauty about this GLUT 1 and GLUT 3? See, they are in the placenta, right? So if there is low blood glucose level, placenta has a higher priority to get the glucose, right? And see, neurons and the brain. Remember this. Don't get confused between this. Brain has GLUT 1, but neuron don't have GLUT 1, but neurons have GLUT 3. If they are very tricky in the exam, they may trick you with that, right? Now, GLUT4 is found in the heart, adipose tissues, as well as the skeletal muscle. Another point to remember, very important point to remember, and that is insulin stimulated glucose uptake. In the skeletal muscles, while doing the exercise, there is an elevated level of the calcium, right? That also triggers the insulin release, and that will lead to the uptake of the glucose. Another point, GLUT2. GLUT2 works opposite to the GLUT4. GLUT2 is there in the pancreatic beta cell, right? These are the beta cells which are gonna release the insulin, right? So when GLUT2 is taking the glucose, it means there are very, very high levels. See, the KM is very high, right? So they will open up only when there is very high glucose available. Too much of the glucose is available, that is during the after the fed state, right? Then GLUT2 will be opening up. They are going to do lots of cascade of events which we have discussed in detail in the class, right? In SWT also for the NEET PG, right? So then they are going to lead to the release of insulin. GLUT2 is helping to release the insulin. GLUT4 is activated when the insulin is there. Okay, so don't get confused between these two. Or we can say after the overnight fasting, there will be decreased GLUT2, because overnight fasting will lead to the decreased insulin level, right? And the GLUT4 will be stored inside, right, in the vesicle mound membrane. And as soon as insulin is available, they will come on the surface to uptake the glucose, right? Which of the following is present in skeletal muscle? We just have discussed GLUT4 is present in the skeletal muscles. There are other GLUTs also, not so important. Just remember this one, GLUT5, that is a fructose transporter. F5 for fructose, right? That is a fructose transporter. Gluconeogenesis is inhibited by. Gluconeogenesis, we are synthesizing the glucose, that is in the starvation condition or prolonged fasting or fasting state. We can say the fasting is the best appropriate word we should be using. So that is initiated by the glucagon, but it will be inhibited by the insulin. Cholecytokinin does not interfere with the gluconeogenesis. It is just helping to improve the digestion by slowing down the emptying food from the stomach. Another function of the cholecytokinin is it stimulates the bile acid production in the liver as well as in the gallbladder. Alpha reductase also has nothing to do with the gluconeogenesis. It processes the androgens. Mainly it converts the testosterone into 5-hydroxytestosterone. Question number 40, very, very important for the glycogen storage disorder. Again, a high yield topic. I'm going to quickly do it. Then we will wrap this session up. Enzyme deficiency in the von Gerke diseases, right? We talk about so many of the glycogen storage disease, 1, 2, 3, 4, 5. 3, Cori, right? Cori is D. Cori's disease is D branching enzyme. 4 is A, that is Anderson is the branching enzyme. So A, B, C, D. Type 1 is the von Gerke disease. Von Gerke disease enzyme deficiency is the just write it down. Maybe next session I'll do this in detail. Okay. So this is, just remember, von Gerke diseases, there is a glucose 6-phosphatase. That is the last step of the gluconeogenesis. When you have to convert glucose 6-phosphate to glucose that can be transported out in the lumen. If that enzyme is deficient, that is going to lead to the von Gerke disorders, right? And the Cori, C, D, this is a D branching enzyme and this is the branching enzyme. So A, B, C, D, remember this, O, B, C, D. Pompey's disease, another very important question they have asked before also, is an acid maltase deficiency, and that is more of a lysosomal storage disorder. So you may study it under the glycogen storage or maybe the lysosomal storage disorders. Type 5 and type 6 are important to pay attention. They have glycogen phosphorylase. Enzyme is same. Right, enzyme is same. Glycogen phosphorylase is there for the glycogenolysis. We are breaking down the glycogen, and glycogen stores are at two places, the liver as well as the muscle. if the glycogen phosphorylase is there in the liver, in the right, in the hepatic system, that is, it is going to degrade the glycogen, that is going to her hepatic, that is liver, is her Her's disease, right? And when the glycogenolysis is taking place in muscles, then it will going to lead to the McArdle disease, that is muscle cramping and all those sort of features you will be seeing, right? So did I tell you the answer? Von Gerke's disease is choice A. Right. Perfect. Next question is enzyme deficiency in Pompe's disease, right? We just discussed, that is acid maltase. Synovial fluid contains what? Synovial fluid contains the cushioning thing, that is hyaluronic acid. This is the chart, I have already given it to you. Please make sure you make this chart down and you go through the chart again, right? Synovial fluid here is there, right? It serves as the lubricant, right? All are rate limiting enzymes except HMG-CoA reductase is the rate limiting enzyme in the cholesterol synthesis. Phosphofructase kinase 1 PFK1 is in the glycolysis. Acetyl coenzyme A, ACC, you have to know the short forms of the enzymes. PFK1, HMG-CoA, right? ACC is the rate limiting enzyme of the fatty acid synthesis. So malonyl dehydrogenase is not a rate limiting enzyme, that is choice D. Again question, but different choices. All are rate limiting enzyme. PFK1, glycogen synthase, phosphorylase, all these are rate limiting enzyme, not the phosphatase. And this is the table I have for you for all the carb metabolism. Glycolysis, which is also known as EMP. Another important thing you should be knowing. You should be knowing that all the names of the pathways. EMP is Embeden-Meyerhof pathway, also known as the glycolysis, right? Location is cytosol and the rate limiting enzyme is PFK1, right? Glycogenolysis enzyme is phosphorylase enzyme, not the phosphatase enzyme. So hence we are eliminating the choice D. Fructose intolerance is due to deficiency of, I'm going to cut this short now, FBI, remember, FBI, right? When we digest the fructose, there is an enzyme that is aldolase. We know that, right? Aldolase. There are two kinds of aldolase, aldolase A, aldolase B. Aldolase A we see in the glycolysis. Aldolase B is for the fructose. So remember this. So fructose intolerance is fructose. This is the way I memorized it myself. Fructose intolerance is what? Is FBI, that is aldolase B. Fructokinase deficiency is going to give you what? Fructokinase deficiency is not like a big deal, but the big deal is hereditary fructose intolerance, right? So fructokinase is giving you the essential fructose urea. Aldolase B is responsible for hereditary fructose intolerance. And another important thing is, over the time patient develops cataract. So what do we see? We see a sunflower cataract, and these are the various images. Where is in the galactosemia, classical galactosemia, again we see the cataract, but that would be oil drop cataract. That will be oil drop cataract. Next is the question about the galactosemia. Which enzyme deficiency causes galactosemia? That is galactose 1-phosphate uridyl transferase, right? This enzyme is deficient. So you have the galactose in the diet. That galactose is coming from the lactose. Lactose is broken down into the glucose and the galactose. Though glucose and the galactose, they are apimers of each other, it is hard to convert them as such. There is a mechanism to it. First, galactokinase will convert galactose into galactose 1-phosphate. So it is staying inside the cell, it is not going out. So just to trap it, for that we need the enzyme galactokinase, right? And then there is this uridyl transferase enzyme. What this uridyl transferase enzyme is doing is, you have the UDP glue. So they are going to exchange the partners. They are going to switching their partners. So this glucose will be exchanged for this galactose. So what we will be having? Glucose 1-phosphate rather than the galactose 1-phosphate. And that this transfer, this swapping of the girlfriends are done by the uridyl transferase. If this enzyme is deficient, there will be increased level of galactose 1-phosphate, which will convert into the galactitol, right? Which will convert into the galactitol, leading to the cataract. And what kind of a cataract? That cataract will be oil dropped cataract. And who is doing it? See, galactose is what? Galactose is an aldehyde. Like glucose, galactose is an aldehyde. And galactitol, look at the name of the enzyme, name of the word. This is an aldehyde, this is an aldol, right? So aldehyde to alcohol conversion is what? Is a reduction process. That is why this is aldose reductase is the enzyme. And what kind of? Oil drop cataract. Whereas in the fructose we saw sunflower cataract. Okay. Reduced form of NADPH. NADPH is produced by HMP pathway, also known as pentose phosphate pathway. These, right? Where it is taking place? In the cytosol. The rate limiting enzyme is G6PD, right? There is no ATP production, and that there are two products, NADPH and ribose 5-phosphate. Ribose 5-phosphate is used in the synthesis of the DNA and the RNA. NADPH is used at so many places for the fatty acid synthesis, lipid, cholesterol, steroid hormones, everything related to the lipid. That is the first half, right? And also for the neutrophils, and also the RBCs make use of the NADPH, right? Reduced form of NADH in citric acid cycle is produced by enzymes. There are 4 dehydrogenases in the citric acid cycle. I have told you before that also. All those produces either NADH or FADH2, right? So these are the 4 enzymes: isocitrate, alpha ketoglutarate, succinate dehydrogenase. And as I have told you, in the electron transport chain also, succinate dehydrogenase has everything to do with the FADH2. So this is the part where you are making the FADH2. Where citric acid cycle is taking place? In the mitochondria. What are the other name of the citric acid cycle? Tricarboxylic acid cycle. Rate limiting enzyme is what? What is the rate limiting enzyme of the citric acid cycle? You have to tell me this. How much ATP is gained? 10 ATP is gained, right? Isocitrate dehydrogenase, alpha ketoglutarate dehydrogenase, malate dehydrogenase, all these 3 will give you NADH. Who can tell me rate limiting enzyme? Rate limiting enzyme is isocitrate dehydrogenase is the rate limiting enzyme, right? Is the rate limiting enzyme. And I thank you all. I have taken too much time, but still we will continue if you like it. And I know there are questions I could not take, queries I could not take. Definitely I'm going to take it tomorrow. Bye-bye. Good luck.